Welcome to all. Myself Sajithra, lecturer of Emmanuel Arasar College of Pharmacy. In this session, I am going to discuss about glycogen metabolic pathway. First of all, glycogen is the storage form of glucose in animals and as starch in plants. In liver, it contains 6 to 8 percentage, and in muscle, it is stored in 1 to 2 percentage. Glycogen stored in muscles are 250 gram and liver about 75 gram. Glycogen functions. It maintains the blood glucose levels. Liver glycogen stores increase in a well-fed state which are depleted during the fasting. Muscle glycogen serves as a fuel reserve for the supply of ATP during muscle contraction. And this glycogen metabolic pathway are classified as two ways, that is glycogenesis and glyconeolysis. Genesis means synthesis of glycogen and glyconeolysis means breakdown of glycogen. First of all, glycogenesis means synthesis of glycogen from glucose, which is known as glycogenesis. It takes place in cytosol and it requires ATP and UTP. ATP means adenine triphosphate and UTP means uridine triphosphate. Synthesis of first steps for this glycogenesis is synthesis of UDP, glucose. Glucose is converted into glucose 6 phosphate in the enzyme hexokinase. It happens in the muscle, and in the presence of glucokinase, this conversion happens in liver. Phosphoglucomutase catalyzes the conversion of glucose 6 phosphate to glucose 1 phosphate. Then uridine diphosphate glucose is synthesized from glucose 1 phosphate and uridine triphosphate by uridine diphosphate glucose phosphorylase. Second step is a requirement of primer to initiate glycogenesis. A small fragment of pre-existing glycogen must act as a primer to initiate glycogen synthesis. In the absence of glycogen primer, a specific protein, namely glycogenin, can accept glucose from UDPG. The hydroxyl group of the amino acid, that is tyrosine of glycogenin, is the site at which the initial glucose unit is attached. The enzyme glycogen initiates synthesis. The synthase transfers the first molecule of glucose to glycogenin. Third step of glycogenesis is glycogen synthesized by glycogen synthase. Glycogen synthase is responsible for the formation of 1,4 glycosidic linkage. This enzyme transfers the glucose from UDP glucose to the non-reducing end of glycogen to form alpha 1,4 linkage. Formation of branches in glycogen. Glycogen synthase can catalyze the synthesis of a linear unbranched molecule with 1,4 alpha glycosidic linkages. Glycogen, however, is a branched tree-like structure. The formation of branches is brought about by the action of branching enzyme, namely glucosyl alpha 4,6 transferase. This enzyme transfers a small fragment of 5 to 8 glucose residue from the non-reducing end of a glycogen chain to another glucose residue where it is linked by alpha 1,6 bonds. This leads to the formation of a new non-residue reducing end that is besiding the existing one. Glycogen is further elongated and branched respectively by the enzyme glycogen synthase and glucosyl 4,6 transferase. The overall reaction of glycogen synthesis for the addition of each glucose residue is glucose N plus 2 ATP, which gives glucose N plus 1 plus 2 ADP plus phosphate. that is inorganic phosphate of 2 ATP utilized. One is required for the phosphorylation of glucose, while the other is needed for the conversion of UDP to UTP. Then we see about the glyconeogenesis, that is breakdown of glycogen. The breakdown of stored glycogen in liver and muscle is called as glycogeolysis. The pathway for the synthesis and degradation of glycogen are irreversible. An independent set of enzymes present in the cytosol carry out glycogenolysis. Degradation of glycogen occurs by breakdown of alpha 1, 4 and alpha 1, 6 glycosidic bonds. First steps of glyconeogenesis is action of glycogen phosphorylation. The alpha 1, 4 glycosidic bonds are cleaved sequentially by the enzyme glycogen phosphorylase to yield glucose 1 phosphate. The process called as phosphorylation continues until 4 glucose residues remain on either side of branching points. The glycogen thus formed is known as limit dextrin, which cannot be further degraded by phosphorylase. Glycogen phosphorylase possesses an molecule of pyridoxal phosphate covalently bound to the enzyme. Second one, action of debranching enzyme. The branches of glycogen are cleaved by two enzyme activities present on a single polypeptide called debranching enzyme. Hence, it is a bifunctional enzyme. Glycosyl 4 transferase activity removes a fragment of 3 or 4 glucose residues attached at a branch and transfers them to another chain. Here, one alpha 1, 4 bond is cleaved and the same alpha 1, 4 bond is made, but the places are different. Amino alpha 1, 6 glucosidase breaks the alpha 1, 6 bond at the branch with a single glucose residue and releases a free glucose. The remaining molecule of glycogen is again available for the action of phosphorylase and debranching enzyme to repeat the reaction stated in the 1 and 2. Then formation of glucose 6 phosphate and glucose. Through the combined action of glycogen phosphorylase and debranching enzyme, glucose 1 phosphate and free glucose in a ratio of 8:1 are produced. Glucose 1 phosphate is converted to glucose 6 phosphate by the enzyme phosphoglucomutase. The fate of glucose 6 phosphate depends on the tissue. The liver, the kidney, and the intestine contains the enzyme glucose 6 phosphates that cleaves glucose 6 phosphate to glucose. This enzyme is absent in muscles and brain, hence the glucose cannot be produced from glucose 6 phosphate in these tissues. Therefore, liver is the major glycogen storage organ to provide glucose into circulation to be utilized by various tissues. In the peripheral tissue, glucose 6 phosphate produced by glycogenolysis will be used for glycolysis. It may be noted that glucose 6 phosphate is absent in muscles. Some amount of free glucose is produced in glycogenolysis due to the action of debranching enzyme, that is alpha 1 6 glucosidase activity. I hope you are able to understand this session and thank you for watching.